So, in continuation to our earlier lectures of the plant physiology and metabolism course, in today's lecture we will talk about the sphingolipids. Sphingolipids are the fourth large class of membrane lipids. They share similarities with glycerophospholipids and galactolipids in that they possess a polar head group and two nonpolar tails. However, unlike glycerophospholipids and galactolipids, sphingolipids do not contain glycerol in their structure. Instead, sphingolipids consist of one molecule of long-chain amino alcohol sphingosine, also known as 4-sphingenin, or one of its derivative, along with one molecule of a long-chain fatty acids. The polar head group of sphingolipids is connected to the molecule either by glycosidic linkage in some cases or a phosphodiester linkage in others. So, carbons C1, C2 and C3 of the sphingolipid molecule correspond to the 3 carbons of glycerol in glycerophospholipids. When a fatty acid is linked to the ONH2 group on C2 through an amide bond, the resulting compound is known as ceramide. Ceramide exhibit structural similarities to diacylglycerol and serves as the foundational structure for all sphingolipids. So, there are 3 subclasses of sphingolipids that all derivatives of ceramide but differing in their groups. They are the Sphingomyelins, neutral glycolipids and Gangliosides. This figure shows the different sphingolipids. The first 3 carbons at the polar end of the sphingolipids are analogs to the 3 carbons of glycerol in glycerophospholipids. The amino group at C2 level bears a fatty acid in amide linkage. The fatty acid is usually saturated or monosaturated with 16, 18, 22 or 24 carbon atoms. Standard symbols for sugars are used in this figure. First, we will discuss about the sphingomyelins, which are categorized as phospholipid because they contain phosphocholine or phosphoethanolamine as their polar head group, akin to glycerophospholipids. They share similarities with phosphatidylcholines in their general properties, 3-dimensional structure and lack of net charge on their head groups. Sphingomyelins are found in the plasma membranes of animal cells, particularly in myelin, a membrane covering that envelops and insulates the axons of certain neurons, hence their name is sphingomyelins. This figure shows the molecular structure of 2 types of membrane lipids, which are the phosphatidylcholine and the sphingomyelin. Next one is the glycosphingolipids. They are predominantly located on the outer surface of plasma membrane. Feature head group composed of one or more sugars directly attached to the OH group at C1 of the ceramide moiety. Unlike glycerophospholipids, they do not contain phosphate. Cerebrocytes consist of a single sugar linked to ceramide. Those containing galactose are typically found in the plasma membrane of neural tissue cells, while those with glucose are prevalent in the plasma membranes of non-neural tissue cells. Globosides, on the other hand, are glycosphingolipids containing two or more sugars, commonly D-glucose, D-galactose or N-acetyl D-galactosamine. Cerebrocytes and globosides are often referred to as neutral glycolipids since they do not carry any charge at the pH 7. Next one is gangliosides. Gangliosides, the most intricate sphingolipids, feature oligosaccharides as their polar head groups and one or more N-acetylnuraminic acid residue, commonly known as sialic acid, at their termini. The presence of sialic acid imparts a negative charge to gangliosides at pH 7, setting them apart from globosides. In plants, two primary types of complex sphingolipids have been identified, that is glucosylceramides or glucocerebrosides and glycosilynositol phosphoceramides. Sphingolipids play a significant roles as major constituent of endomembranes, with approximately 40% of the plasma membrane containing sphingolipids. They are also found in the tonoplasm. These sphingolipids are synthesized within the endoplasmic reticulum and Golgi apparatus. In plants, sphingolipids are implicated in programmed cell death associated with defense mechanism. This slide discusses about the sphingolipids at cell surface, that is basically the site of biological recognition. Discovered over a century ago by John Todichum, sphingolipids initially posed a biological mystery, leading Todichum to dub them after the enigmatic Sphinx. In human cellular membranes, more than 60 distinct sphingolipids have been identified, with many being particularly prevalent in the plasma membranes of neurons. While some sphingolipids serve as a recognition sites, on cell surfaces. Only a few have had their specific functions elucidated thus far. The carbohydrate components of certain sphingolipids determine human blood group types, affecting blood transfusion compatibility. Gangliosides concentrated on cell exteriors aid in recognizing external molecules or neighboring cell surfaces. During embryonic development, the composition and quantities of gangliosides in the plasma membrane undergo significant shift. Tumor growth prompts the synthesis of new gangliosides, and specific ones have been found to induce differentiation in cultural neuronal tumor cells even at low concentrations. The exploration of gangliosides, that is the diverse biological role, remains an active area for the research. Most cells undergo continuous degradation and renewal of their membrane lipids. So, each hydrolyzable bond in glycerophospholipid correspond to a specific hydrolytic enzyme present in the lysosome. Phospholipases of type A are responsible for removing one of the two amino acids, resulting in the production of lysophospholipid. It's important to note that these esterases do not target the ether link found in plasmalogens. Lysophospholipases then eliminate the remaining fatty acids. Gangliosides, on the other hand, are broken down by a series of lysosomal enzymes that sequentially remove sugar units, ultimately yielding a ceramide. Any genetic effect in these hydrolytic enzymes can lead to the accumulation of gangliosides within the cell, resulting in several medical complications. Phospholipases exhibit specificities in their actions. So, phospholipases A1 and A2 target intact glycerophospholipids, hydrolyzing the ester bonds at C1 and C2 of glycerol respectively. Phospholipases C and D, on the other hand, each cleave one of the phosphodiester bond within the head group, while certain phospholipases are selective and act only on particular types of glycerophospholipids like phosphatidyl inositol 4,5 bisphosphate or phosphatidyl choline. Others demonstrate broader substrate specificity as shown in the figure. The moving to next is the sterols, which are a class of membrane lipids characterized by their unique structure composed of 4 fused rings. 3 of these rings contain 6 carbons each, with the fourth ring consists of 5 carbons. The presence of a hydroxyl group at the third carbon position in ring A renders one end of the sterol molecule polar, while a hydrocarbon chain at carbon 17 makes the opposite end nonpolar. Consequently, sterols exhibit amphipathic properties, positioning themselves within the lipid bilayer of membranes with their polar hydrophilic end facing the aqueous environments. Sterols play a crucial role in determining the properties of membrane. In plant cells, cholesterol, a common sterol found in animal cells, is absent. Instead, various other sterols such as sterol are prevalent in plant membrane, including those of mitochondria, endoplasmic reticulum, and the plasma membrane. Fungi, on the other hand, contain ergosterol as a major sterol component in their membranes. These sterols differ in their hydrocarbon chain attached to carbon 17 of the styrene nucleus within the molecule's structure. Interestingly, bacteria lack the ability to synthesize sterols, making them distinct from eukaryotic organisms in terms of membrane composition. Instead, bacteria utilize alternative lipid components to fulfill similar structural and functional roles within their membrane. The cholesterol is depicted in a chemical structure where the rings are identified as A through the D, facilitating the discussion of its derivative as shown in this figure. The carbon atoms within the structure and numbered in blue for clarity. The C3 hydroxyl group highlighted in blue shading as shown in the figure constitute the polar head group of cholesterol. To facilitate storage and transport of the sterol, this hydroxyl group combines with the fatty acid to create a sterol ester. Here we will discuss about the extracellular lipids. The cuticle is a hydrophobic layer found on the surface of leaves, serving to minimize water loss and offering protection against pathogens while providing structural support to plants. It comprises two main components, that is cutin and cuticular wax. Cutin is a characteristic feature of plant cells located exterior to the epidermal cells of leaves and integrated with the cell wall. Produced by non-photosynthetic epidermal cells, cutin is polymeric molecule primarily composed of oxygenated fatty acids with carbon chain length of 16 or 18. These fatty acids form monomers that can be monohydroxy, polyhydroxy or epoxy in nature. They create ester linkages with glycerol or with primary or secondary hydroxyl groups present in other fatty acids, resulting in the formation of an inelastic and hydrophobic framework. This structure allows for the passage of water from the leaf surface due to spaces between molecules, which are filled by cuticular wax. Pathogen seeking entry into plant cells must produce cutinase enzymes to degrade the cuticle and facilitate their invasion. Now here we will discuss the another extracellular lipid, that is suberin. Suberin is an extracellular essential lipid found in plants, differed from cuticle in its fatty acids composition and structure. While both are polymeric molecules, suberin's fatty acids feature longer carbon chains, typically ranging from 18 to 22 carbons compared to cutin. Unlike cutin, the fatty acids in suberin lack oxygenation, secondary alcohol or epoxy groups. Moreover, suberin contains a notable amount of dicarboxylic acids and phenylpropanoids, which are partially linked together similar to lignin. In terms of deposition, suberin is typically found towards the inner side of the primary cell wall, whereas cutin is deposited towards the outside of the epidermal cell wall. Additionally, the deposition of cutin is polar, occurring only on one side of the epidermal cells, while suberin is generally distributed around the plasma membrane of cells. The Casparian strips found in the epidermis are composed of suberized cells, rendering the cell wall impermeable to water. Consequently, water and minerals cannot pass through the apoplast to reach the plant vasculature. Instead, they must traverse the symplast beyond the endodermis in roots. This mechanism prevents water and minerals from diffusing back from the plant's vasculature, thus contributing to root pressure. In C4 plants, bundle sheath cells contain suberin depositions in their cell walls, making them impermeable to carbon dioxide. Suberin is also present in cork tissue consisting of diseased cells enveloped by alternating layers of suberin and wax. It is deposited in response to abiotic stress and mechanical injury. The waxy layer on pollen grains plays a crucial role in pollen pistil interactions known as exine, which contains other lipids embedded within it. Exine is composed of a complex polymer called sporopollenin, similar in its structure of the cutin.